And our goal here is to construct a all-item model of the target sequence without a template structure. So you can see from the from here, it goes from a random conformation and minimize the energy until we reach to the final predictive structure. There are two major components for temperate-free modeling. Search algorithm, which is an efficient confirmation sampling algorithm to explore the huge confirmation space. The evaluation function is an accurate energy function to differentiate the native state from decoys. We need to tell which decoys is the final result very near to the native states. And also, there are two subcategories of the temperate-free modeling. Physical modeling simulates the physical procedure of protein folding using microdynamics. And statistical modeling based on the combination of, the predictions are made using a combination of the statistical models relating the sequence to structure. And it's typically mined from a database of structures. So our focus is on the statistical modeling. There are some leading methods now. The representatives of them are lattice model, which the best representative for this method is the touchstone stone, and also fragment assembly by Rosetta. And the mixture of lattice model and the fragment assembly model is by TASA. One common feature of those leading methods is that they all use a discrete representation of a protein conformation so that they can dramatically reduce the search space. Although very successful, they have some major issues. One of them is that the protein structure space is continuous. So even a small change in the backbone angles can result in a very different conformation. Another thing is that, well, as mentioned by Andreas Fieser on the 3D talk earlier this week, the new folds actually seems to be composed of very rarely occurring supersecondary structure motifs. So to address those issues and start to solve them, people have proposed to sample in a continuous conformation space. CRF folder is previously done by our group, and also like Hammerich from Copenhagen and Yauchi from Indiana, they both do something other methods, but they are all done with the continuous conformation space. I'm going to talk a little bit about the CRF folder we have done before. CRF folder sample protein conformations in a continuous space without using any fragments. Currently, it's as efficient as those leading methods we mentioned. CRF folder efficiently sampling backbone angles using conditional random fields from protein sequence. More detail about this work can be found in our paper in 2008 and 2009. There's a couple of things that I want to address here about CRF folder. First one is that CRF folder uses a very simple energy function with only 3 terms. Another one is that we only use the main chain and the C beta items without building the side chain. So that reduces the computation time dramatically, while also keeps the accuracy. The performance of CRF folder is comparably to those leading methods, for example, Sagani's TASA and Baker's Rosetta. Today, I'm going to present you with our new work, the CNF folder. This CNF folder is based on a recently invented probabilistic graphic model, conditional neural fields, also from our group, For more detail, you can refer to the paper Conditional Neural Fields from NIPS 2009 by Jen and etc. This conditional neural field is a perfect integration of neural field, neural network, and the CRF. With the CNF folder, we can actually model the nonlinear relationship between sequence profile and the structure, while CRF folder cannot. This CNF folder is coupled with a simple energy function as well, but use a different energy minimization methods. We use replex exchange here. Coupled with replex exchange, Monte Carlo simulation, CNF folder generates much better decoys than CR folder. Our CR folder and the CNF folder both use continuous representation of the backbone. Here, I want to address again about this. We only use main chain and CRF items. So, firstly, we represent the backbone using CRF items. Each node in this picture is represents a CRF item. We use theta and the tau angle. Theta is a virtual bond angle between neighboring virtual bond composed by CRF items. And the tau is the dihedral angle of the neighboring the hyperplanes. The distribution of theta tau pair, you can see from the second graph. Once we have the CRF items in the backbone, we can add the other main chain and the C beta items using the extended BBQ method. The theta tau pair we use in the CNF folder and CF folder actually is those backbone angles clustered into 100 groups, and each group is modeled by a 5-parameter Fisher-Bingham distribution. You can see this distribution more clearly from this graph. Actually, the angle pair groups in right happens mostly in alpha helixes. Similar for blue in beta strand regions and green in loop regions. So now I'm going to talk a little bit more about this CNF model. For given input sequence of sequence profile and the second structure informations, we have input feature with a window size of 9. We want to predict the backbone angles. So each ISI here represents a FEP5 distribution at the position I. So for ISI, we use a window of 9 with input sequence features. CRF links each of the input to each of the backbone angle. You can see here it's a linear combination. The CNF add a hidden layer of neurons. So each of the input links to is a linear combination input feed into the neuron, and every neuron have a output feed into the backbone angle at each position. More formally, the CNF model, here we use the second order CNF model. The CNF model aims to maximize the conditional probability given of the backbone angles given a sequence profile. So f here is the feature function at each position. It's composed of edge features, which describes the relationship between neighboring torch angles. And state features is used to describe the relationship between the sequence profile and the backbone angle. So you can see that from here is a linear combination of neurons, and that's the function of neuron gates. The CNF model is trained using around 3,000 non-redundant proteins with less than 30% sequence identity and resolution better than 2 Armstrong. We use same training data as what we did for CRF folder so that we can compare the performance of those two models. And also, those data actually are prepared before cosplay so that we can use cosplay targets as a testbed. For validation, we use 5-fold cross-validation, and we regularize the model to avoid overfitting. But by the way, we also, for validation and test targets, we also try to avoid those targets with big sequence identity with the training data. Eventually, we choose those models with biggest conditional probability on the validation data set. Eventually, the CNF model we get regarding to this conditional probability is actually much better than the CRF model. Talking about the probability, CNF model achieves a probability better in magnitude of over order of 10 than the CRF model. So once we have this CNF folder, the model is trained, and we can then sample the conformation. Given this sequence and the initial conformation, pretty random. Firstly, we sample a segment with size from 1 to 15. And then we sample the backbone angles out of this segment by the probabilities from the CNF model. So each ISI here, as we mentioned, is a FB5 label. When we have the label, we use the IP5 distribution to sample the real valued angles of theta tau pair. And then we can reveal the coordinates for the CRF items because we have the theta pair and also the fixed band length. Eventually, we reveal the other main chain items from CRF to get this ribbon. The energy function we used has 3 terms. Dope is distance-dependent pairwise statistical potential. ESP is a simplified solvent-accessible surface area potential. KMB bond is Baker's hydrogen bonding energy for beta forming. The energy minimization method we use here is rapid exchange. In our experimentation, we have used 20 replicas with temperature from 5 degrees to 100 degrees. And between each conformation swap, we sample 30 steps. Those actually, each step is described in the former slides as shown. After each 30 sampling steps, we have a conformation swapping. We swap neighboring replicas based on the probability relating to their energy difference and their temperatures. In total, we have 800 exchanges from the start configuration until we reach to the final configuration to get the stop structures. And those structures in the stop configuration are actually the final decoys we're going to generate from this CNF folder. So now we can see some results. The performance of CNF folder is significantly better than the CF folder. Here is the x-axis is denotes the energy, and the y-axis denotes the RMSD. So you can actually see here from here, those blue regions, which represents the decoys generated by CNF, has much lower energy than the CF folder. This side is smaller energy. And to compare with the other groups, we have a table here, lists the results of a clustering result comparing with the cost-based remodeling targets. So for the decoys generated, we cluster them into 5. And this area is the results of our first cluster centroid comparing with the first model submitted by each server from Cospate. And this area is the is our best cluster center out of 5 cluster center, comparing with the best server submissions from other groups. So you can see from here, those right targets, we did very, very good. Out of the second number is the number of targets submitted by the other groups, and the first number is our rank in that submissions. So those are the best in all the groups. And the green, blue, you can see that it's very good. And those blacks are fairly good. One thing to notice here is TO498D1 and TO410D3 are the truly new folds for Cospate, and we did very, very good on those two. And you can see better from those 2 charts here, and this is our result. The best one, the first one, and those are the server, the results submitted from the other servers. So you can see that we are really far above the other guys. And here, this is our couple of superimposed structure of our of those 2 targets. We don't generate a lot of decoys. For this one, we achieve 5 Armstrong, which is it's a pretty long sequence, and we only use 9,000 decoys. And for TO510D3, we generate only 6,000 decoys, and this RMSD is 2.99 Armstrong. So for conclusion, the CNF model can capture the nonlinear relationships between the protein sequence and the structure. And CNF folder can minimize energy function to a lower values and clearly outperforms the CRF method on most of the target test proteins. Again, although we only use very simple energy function and only see beta representation for the side chains, CNF folder exhibits advantage on mainly alpha and small beta proteins. The next thing we want to do to CNF method is to model long-range hydrogen bonding so that we can improve the longer beta proteins. We have a poster at the area of W48. Please come by if you have further questions. Eventually, thanks for the funding from TTIC Research and computing resource from Temperate, SharkNet, and OSG, and also thanks for ISCB, DOE, and NSF for supporting our travel fellowship. Thanks.
So the rationale for using fragments in Rosetta is that there are local interactions between I to I plus 1, I to I plus 2, I to I plus 3 that are not very well understood by the force fields, not very well modeled by the force fields. And so by using fragments from the database, you get things that are realistic on that level. And that information seems not to be included in your methodology. So is it in there somewhere that I missed, or is that something that you could additionally add to improve things, or?
As you mentioned before in the former slides, you can see that those fragment-based methods, they build the fragments, but those fragments actually, you can see that the new fields, they don't have exact the same as those in the fragment library.
I would like to answer this question.
Okay.
So actually in our model, we do consider the interaction, the local interactions so in 2 items. So if you still remember our CNF models, in our CNF model, we consider the dependency among 3 adjacent residues. So in this, we have one edge feature. So edge feature will model the local interaction among 3 adjacent residues. The second item is we predict the background angles, and the background angles depend on a window of sequence information. For example, the background angles at each position depend on the residue information in a window of 9. So actually in our model, we model the local interactions, but in a different way than the fragment-based method.
And this representation actually is more close to the real situation.
Can you briefly comment on the table with targets? So you have overall seem to be very good results, but 2 targets were rather bad. So did you check what those structures, what make them so different?
Okay. I think actually you can see from here, those 2 targets, this one and that one. The reason why it's not so good is because of the energy. Although we minimize the energy to a much lower range, but this energy function is not very good for those 2. So you can see that lower energy doesn't really mean lower RMSD.
But when you look on the actual structure when it's got published, can you, I don't know, comment what was so specific there? Is that many unstructured loops there, or why your method fails?
We only observe the energy difference between the energy function, the difference of the correlation between energy and RMSD there. For the physical mechanism, we are not clear yet.
One more question on the testing. When you were doing the testing on I think you reported a lot of results on single domains that were part of multi-domain proteins. Did you do the testing just on those domains, or did you test on the full protein and then extract the domain? Because what everyone else was doing was predicting for the whole protein. So comparing small domain where you did just optimization of that domain after knowing what the domain was is quite a different problem.
We only compare on the domains instead of the whole.
Actually, we use the domain cut in Cascade, so the domain boundary is incorrect.
It's very difficult to get that
We use
domain
the correct domain boundary. We use the wrong domain boundary in Foley. In our experiment.
You use the predicted domain boundary?
Right, we use the predicted domain boundary in Cascade. We don't use the correct domain boundary.
few minutes to change the